about the Creation Summit. This is the first one of these events. Now the folks that have come together, it's kind of a loose association of lots of small people. Well, that's not a very good way to put it. Not huge ministries like Answers in Genesis, but folks that God's put it on their heart and they're involved in science and they're involved in creation teaching. And there's about 20 of us on this board of directors for Creation Summit with one purpose only: to bring credible conferences and speakers onto major college campuses. And we're hoping to do 2 of them per year. And the very first one that's ever going to happen is right here in Lansing, Michigan, next November. It's the idea of reaching the next generation with the truth of God's Word. And there's a verse that's been put on my heart that I've just used over and over again over the last few years. And that's what I believe to be the Great Commission in the Old Testament. See, God's commands haven't changed. God says, I changeth not. And this is a statement God made in the Old Testament. He said, this is our primary job. One generation shall praise the works of God to the next generation. We shall declare God's mighty acts. I'm going to talk about that in a second, and I want you to visualize what our world would look like if we did nothing but that. And by the way, that statement was made before Jesus came to take the sins that we brought upon ourselves upon him himself. Before that ever happened, this statement was made. And we'll just take a quick look at what are these great acts, great works of God, his mighty acts. Now here's the method of the Creation Summit organization: to put conferences on major college campuses that connect the real world with the Bible. Because you know that's not what's going on. Students, whether they come out of a Christian church or not, they look at the Bible as a book of religious rules and religious philosophical concepts, but they don't look at it as something that is real to real history, real science, real stuff going on around us. And we've got to change that. They've got to change their way of thinking. Jesus said the same thing. In John 3, verse 12, he said, if I've told you of earthly things and ye believe not, how shall you believe if I tell you of heavenly things? See what God did when he came to earth? He connected everything dealing with the physical world with the stuff dealing with spiritual concepts, salvation, heaven, hell, all that. He never disconnected them. Later on, and I didn't throw up the quote, Jesus said, if you don't believe Moses and the prophets, you won't believe it even though someone rise from the dead. See how he's connecting real stuff, real history. What did Moses write about? Biology. Where did everything come from? Animals reproduce after their own kind. Geology, a worldwide flood. The entrance of death into creation. Real events, real history, real science, reality with spiritual stuff. What he's saying is if the Bible's wrong about that stuff, why would you expect people to believe it when it talks about other stuff? This is so foundational, what we're trying to do. So we're going to bring in credible, interesting, absolutely phenomenal, world-class experts onto these college campuses. Now, I don't even fall into that category. I'm a research leader for Dow Chemical. I spent my whole life doing science, 18 patents, multi-million-dollar product line. But if I come onto a college campus, I don't have a PhD. My degree is chemical engineering. I'm going to be immediately disregarded because I don't have the credentials people think are needed to speak upon these subjects. So a little later, I'm going to show you the kind of people we're going to bring in. It has to have local support. It has to have publicity. Part of the cost of this conference, we are going to have brochures that describe these very interesting classes that are going to be taught. It's going to be a one-day seminar on a Saturday morning to Saturday late afternoon. There's going to be multiple sessions going on simultaneously. It's going to have a little biography of these speakers from major universities, and it's going to talk about the relevance of it all. But we've got to get the Christian groups on campus pumped up and passing these out and talking about it and bringing it into their classrooms. It's why when we talked to Larry, he said, don't do this in the spring. Come to our groups, lay the groundwork, get the students excited in the fall, and then get them as an army out there on campus to get the other people around them excited. So that's the goal. Do it well, do it right. And lastly, high quality resources left with the students. Because if they just hear it, then they're going to be inundated by the other viewpoints. So you got to leave them with some resources. That's why we're going to leave these books with the students so they can follow up. Now these are the mighty acts of God. These are the things left out of their education. And you think about our world, if in our colleges, alongside of this idea that one animal turned into another, that turned into another, that turned into another, that the other alternative is there is a creator who made very different groupings of creatures sometime in the past. Totally different viewpoint. And here's the evidence that supports it. There's a creator that made the DNA code. Here's the reason the DNA code couldn't have made itself. But they don't get to hear any of that. Imagine our world if students in universities were taught, you know, the reason death exists is because we rebelled against the one who made us. And had he allowed us to live forever, we would have been forever separated from him. Death exists as we rebelled against the one who made us. And he didn't want us to be forever separated. A real event that really happened in real time, real space, not that far back in history. Not fantasy, reality. All of a sudden they're looking at death in a very, very different way. But how do you put that in this view of world history they've come to be told is reality? You have to explain to them why the rocks exist, why the fossils are there, where continental drift fits in, where does the ice age fit in, where do the dinosaurs fit in. It all fits into the reality that God's Word spends more time talking about a world restructuring, total global, total, every mountain under heaven covered with water, flood, than it does talking about the creation of the entire universe. That's geology. See, the book talks about biology, it talks about history, it talks about why death exists, it talks about geology. And if you get those things wrong, you're going to misinterpret the rocks, you're going to misinterpret the fossils, you're going to misinterpret everything. And then people spread out after this flood, and then in the most momentous event of all human history, the one who made everything entered into creation to take that penalty of death upon himself. But you see, if this is fantasy, then this becomes fantasy in the minds of people too. And more people become to think Christianity is irrelevant. So you just connect it all, and then you show them scientifically why it really is true. That's the purpose of the conference. Now, here's our typical schedule that we'll have in the fall. We'll start at 10 a.m. Hopefully we can get students up that early. We wouldn't think of starting on a Saturday morning earlier. We'll have introductions and orientation, and then probably a half-hour opening address to the whole auditorium. We hope to have an auditorium that'll hold 500-600 people. Then we'll break into workshop sessions where we're going to have 4 major speakers, each giving a different topic. One on genetics or biology, one on geology, one on cosmology, time, space, energy, matter, physics, and I think genetics would be the third one, all right? So students can pick which session they want to go to. Lunch break, then another set of workshop sessions with another set of very relevant, interesting topics, wrapping up with a panel discussion where students, and by the way, each of these are an hour and a half so they can ask questions of these expert speakers. Wrapping up with a panel discussion, and we're hoping to have a major debate in the evening, but that hasn't come together yet. We'll see whether that pulls together or not. It's still pending. So that's the idea. Now, in 5 more minutes, I want to start to give you an introduction to the kind of things that are going to be presented to these kids. And then Dr. Wile is going to take over with some of the latest phenomenal finds that support a biblical worldview. Whenever I go onto a college campus, I don't start preaching the Bible. I just treat people with respect, understand that they've been taught a certain viewpoint, and start helping them be logical. First of all, acknowledge there are 2 possibilities for our existence. It's like if you can envision the entire universe is represented by this box, all right? All the stars, all the energy, all the time, matter, space, and inside the box you find these fine jewels and pendants and, you know, fine crafted necklaces and stuff. That might be representative of the animals and penguins and giraffes and great whales and rainforests here on Earth. Either the box made itself, that would be natural processes did it. Some process allowed the box to make itself, some process allowed everything in the box to make itself, or A box maker made the box. A box maker made what's in the box. Now, those are the only two possibilities. See, if you want to say, well, maybe God started the process that allowed everything to make itself, you're still saying everything made itself. You're still saying there's no evidence from observing creation that there's a creator because everything can be explained by natural processes. Now, this is why this is so important, folks. That's all these kids get to see here in school. Hour after hour, year, month, decade after decade, in little subtle ways and overt ways, everything in the textbooks is forcing them to think in that direction. And they don't get to hear the alternative. Now, what can science do? See, science can't prove either one of these. I never say that you've proven something with some evidence, because you don't know what you don't know. It's actually a very profound statement if you think about it. You don't know what you don't know. Furthermore, you don't know what you haven't been told. And students in universities, they don't understand what's been left out of their education. Now, here's how science works. It's a classic statement by a man named Dr. Karl Popper. He said, science has to work by being falsifiable. You have to show that something isn't true or it's not science. There has to be a way to show whether it's not true. It's like this. Suppose I wanted to believe all swans are white. Now, a scientific theory or hypothesis is really just an idea. It's really just a belief that you go and find a data and observation to see if it's true or not. But can you ever have enough observations? That's the question. Suppose I went all over Michigan on my summer break, took my notebook, and everywhere I went I saw nothing but white swans, and my hypothesis is all swans are white. Have I proven every swan in the world is white using scientific observation? Okay, this is a chance for interaction. What do you think? No. Absolutely not. But how about if I Facebook my friends who know people in Russia, who know people in Germany, who know people in Australia, who know people in the Polynesian Islands, who know people in Alaska, and they all Facebook and Twitter and write back and forth, and I get 100 million observations back from all over the planet. And they all tell me they've only seen white swans. Have I now used science to prove all swans are white? No. Still haven't. Because I'd have to be everywhere instantaneously and be a perfect observer and never miss anything. But there is a way to know, using science, whether or not my belief, my hypothesis is correct. And that's if I find a black swan. Now here's what's going on in our public education system in America. There are two possibilities for the box. Either the box made itself, or something outside the box made the box, all right? By law, we have allowed to develop a system in America that says you cannot have a public institution promoting a religious belief, all right? Now, it's selectively applied. I won't even get into that, okay? But if you even cast doubt upon the idea that the box made itself, and by the way, Georgia did this. They passed a law about 10 years ago that said that you have to put a sticker on your biology books that says evolution is a theory, not a fact. The Georgia Supreme Court threw that out and said, by doing that, you're promoting religion. And they couldn't even say it was a theory. Now, they're just being logical because there's only two possibilities. If you cast doubt on the idea that the box didn't make itself, what's the only thing left? The box has a box maker. And that's what's going on. So by law, it gets left out. So the belief that the box made itself is like believing all swans are white. What I'm going to show you is just little bits and pieces later on after Dr. Wile gets done of every area of science where there are these enormous black swans that say the very laws of science show us the box could not possibly have made itself. And it doesn't get shown to the students. So that's what we're going to do in the fall. All of these experts are going to come in and they're going to show these kids the black swans. And they're going to put cracks in their belief system. And they're going to start opening up conversations. And there are going to be lives changed. And I just praise the Lord you guys are here and want this to happen because you're going to be foundational in making it happen. Now, I'm going to have Dr. Wile come up next. My friend, we've worked together. There's just a wonderful gentleman named Pastor Boyd down in Indianapolis. He's got the heart of gold, trying to wake up the church down there. A little disorganized at times in his way he goes about it. He ran Dr. Wile up and down the city like about every Wednesday for 2 years, speaking in dozens and dozens and dozens of churches. But Dr. Wile is an example of who we're bringing. Impeccable credentials. Now, you're not available in the fall, otherwise you would be on the agenda, and there'll be other opportunities. Degree in nuclear chemistry. He is the don't contradict me now. He is the definitive word on Christian science textbooks.
No.
Christian schools, home schools, anybody out there that's trying to bring the box maker viewpoint and the evidence behind it to their children is going to have heard of apology of textbooks. So I thank you for your service, your work, and I can't wait to hear what is the latest, because you always have the latest in creation evidence. Thank you, Dr. Wile.
Are we on? Okay. It's really great to be here. Thanks so much. Got to drive up here today from Anderson, Indiana. I do write textbooks, but I'm not the definitive word in science textbooks for Christians. But I do write textbooks. I've written junior high school textbooks, and I've got an elementary series out now. What I want to talk about today is some of the more recent findings that science has produced that either confirm a creationist view or refute an evolutionist view. I think it's an incredibly exciting time to be a creationist, because there are some monster discoveries out there that really show how strong the creationist view is. If you're interested in any of this, you can find notes on my website. And these notes have all the references to the scientific literature so that you can look in detail if you want. All you have to do is go to my website, click on handouts, and then click on the event listing. And you can like me on Facebook and all that kind of stuff. Okay, so I think really the most exciting thing that's happened probably in the last 2 or 3 years has to do with what was learned just about a year and a half ago about DNA. Since the 1970s, one of the fundamental views of evolutionists when it comes to human genetics is that a large portion of our genome, and the genome is the collection of all the DNA that's in the center of the cell called the nucleus, but the thought has been among evolutionists since the early 70s that the vast majority of the DNA in our genome is junk. It doesn't have a purpose. It's left over from failed attempts at evolution. And this view was so prevalent that back in 1989, when they were trying to figure out exactly how they were going to sequence the human genome and figure out exactly what's in every bit of our DNA, many evolutionists were saying there's no reason to sequence the whole genome because most of it's junk. And so there's no reason to look at it all. As a matter of fact, one of the real big proponents of that was named Sidney Brenner. He was the director of the Molecular Genetics Unit of Britain's Medical Research Council, so he knows a thing or two about genetics. And he said that there's no reason to sequence anything but about 2% of the human genome because the other 98% is simply junk. And this was considered, and it makes sense from an evolutionary viewpoint. If you're counting on mutations filtered by natural selection to produce evolution, you're going to have a lot of failed attempts. And so there's going to be a lot of just nonsense in the genome. More recently, Dr. John C. Advice put it this way: the vast majority of human DNA exists not as functional gene regions of any sort, but instead consists of various classes of repetitive DNA sequences, including the decomposing corpses of deceased structural genes. To the best of current knowledge, many, if not most, of these repetitive elements contribute not one iota to a person's well-being. They are well documented, however, to contribute to many health disorders. And junk DNA has become so foundational in evolutionary view that AVITA, which is sort of the gold standard of computer programs that try to simulate evolution, AVITA will not produce evolution unless 85% of the simulated genome starts out as junk. So it's a fundamental part of evolution. We now know it's utterly false. Just a year and a half ago, there was a timed event where an entire group of studies were all put out at the same time. The studies involved analyzing more than 1,600 data sets, looking at 147 different human cell types. Are you surprised that there are 147 different human cell types? Actually, there are a lot more than that. Throughout the course of your life, from fertilization to death, there are probably about 2,000 different cell types that progress through your body. However, about 200 are really important to stick around for a significant amount of time, and they've studied 147 of them. And there was a whole bunch of stuff learned, but one thing they learned by studying all these gel cell types is this. As the journal Nature says, among the many important results, there is one that stands out above them all. More than 80% of the human genome's components have now been assigned at least one biochemical function. So from the early 70s, we've been told assuredly by evolutionists that the vast majority of the human genome is junk. And it's actually a fundamental part of the mechanism of evolution. All the evolutionary innovation that's supposed to happen in the genetic code is supposed to come from the junk. We now know that the vast majority of the human genome is in fact functional. And please remember, these studies only look at 147 different cell types. As we look at more cell types, we'll probably find that those cell types use part of the genome that other cell types don't. So most likely this 80% will simply go up the more we look at these different cell types. As a matter of fact, Ewan Birney, who is the project's lead analysis coordinator, he calls himself a cat herder, because, I mean, there are literally hundreds of scientists who worked on this project. It's called the ENCODE project. And so he had to herd them all together and coordinate the analysis. He says it's likely that the 80% will go to 100%. We really don't have any large chunks of redundant DNA. This metaphor of junk isn't that useful. So even though we've been taught, remember at university, I was taught definitively that most of the human genome and most of all vertebrate genomes are composed of junk. That's a definitive evolutionary fact. We now know that's utterly wrong. Now, this idea of junk DNA is absolutely necessary in evolutionary views, but the creationist view is exactly the opposite. The creationist view is that DNA is a highly structured, created system, and like a car, a few things might break down, and the car can still go. For example, if you go out and look at my car, I had to tape one of my windows shut because the mechanism that opens and closes it failed, and so now it's always falling down. All right, that doesn't keep my car from going. It's a junk part of my car, but my car can still run. So the human genome could have some junk in it, but just like a car, because the human genome is designed, if too much of it gets destroyed, it won't run anymore. And that's the point here. The vast majority of the human genome we now know is functional. This is perfectly consistent with the creationist view and is diametrically opposed to the evolutionist view. And it's interesting to watch how evolutionists deal with this. There are some who now claim, well, evolutionists never really said junk DNA. And that's nonsense, because all you have to do is point them to the scientific literature to show that. Most of those hardcore evolutionists who have looked at this data try and say that the functions that have been assigned aren't real functions. So as a result, these folks are not defining function properly. But the fact is, the project ENCODE has shown that the vast majority of human DNA is indeed functional. And I think that's very impressive. Over the last few years, we've found a couple more problems with the geological column. Hopefully you all know a little bit about the geological column. Sedimentary rock, the kind of rock that has fossils in it, tends to form layers. And the general view is each layer represents an era of time. And as I dig down through these rocks, I go backwards in time. Well, generally speaking, these rocks tend to contain fossils that are fairly indicative of the layer. So way down at the bottom, you find very simple single-celled creatures. You go a little higher, you find some complex animals, but they're still simple compared to other animals. We call these invertebrates. They're animals without backbones, so you can think of them as like congressmen. So in the slightly higher layers, you have sort of the simple congressmen, and then the congressmen get more advanced. Eventually the vertebrates come along. These are the animals with backbones. They're a little more complicated. They come here. Then we have the land plants, and then reptiles and mammals, and eventually man. All right? And so the idea is, since each one of these layers of rock represents an era of time, we know what lived in these times by what fossils are in the rocks. So for example, we've all been taught that dinosaurs and humans didn't live at the same time. And the reason is we find dinosaur fossils in these layers, but we don't find any human fossils in these layers. Well, since dinosaur fossils are in these layers, this is the era of time where dinosaurs lived. Human fossils are found up here, and no dinosaur fossils are found up here. And so since the fossils aren't found in similar layers, that tells us they didn't live together. And that kind of reasoning is used to tell us a lot about what happened in Earth's past. Now, before I go into the modern problems with the geological column, please note there's nowhere in the world you can go and see this. There's nowhere in the world you can see all the layers with fossils in between and all that kind of stuff. This is sort of a hypothetical construct of what the geological column should look like. All right? But nevertheless, even assuming that this is a real something that's real, the idea that humans and dinosaurs couldn't have lived together because they didn't exist in the same layers has been shown to have some problems. And one of the more recent examples is this one. If you go to a museum that has dinosaur exhibits, you'll see dinosaurs, models of dinosaurs, or their skeletons or something like that. And if you look at what they're walking in, you'll notice they're never walking on grass. And the reason is, dinosaur fossils are found in these layers, but grass fossils don't appear until this tertiary layer up here, and there are no dinosaur fossils up here. So in the end, since dinosaur fossils are down here, and there are no grass fossils are up here, and there are no dinosaur fossils, what does that tell us? Grasses and dinosaurs didn't live at the same time. So it's not surprising then you go to a museum and you find dinosaurs, models of dinosaurs and so forth, but they're not walking on grasses, they're walking on ferns and things like that. For example, if you read You Are Here, A Portable History of the Universe, you'll read this: bats, mice, squirrels, and many aquatic birds, including herons and stork, appear during this period. That's the tertiary period, after the dinosaurs have gone extinct. As do shrews, whales, and all modern fish. All major plants make their appearances, and grasses evolve. Grasses evolve in the tertiary. They weren't around when dinosaurs were around. That's what the fossils tell us. However, believe it or not, there are people who study fossilized dinosaur dung. In order to make it sound a little more appealing, we call it coprolite, but it's fossilized dinosaur dung. And so these people who are very committed to their science analyzed this fossilized dinosaur dung, and guess what they found inside? The remains of grasses. So supposedly dinosaurs and grasses didn't live in the same time period, but dinosaurs ate them anyway, even though they didn't live in the same time period. So obviously this reasoning is false. The first study that came out on this was 2005, and no one wanted to believe it, because the fossil evidence is so clear. Dinosaurs and grasses didn't live during the same time. However, 2011, 6 years later, a much larger study looking at a lot more dinosaur dung, found a lot more evidence for these grasses. So in the end, we now know that that idea is just wrong. Even though they don't appear in the same layer, they must have existed at the same time. What this tells me is those layers don't represent eras of time. They must represent something else, like ecosystems or something like that. Here's the other one that's very interesting. I find this more interesting. It's a little convoluted because it shows you the kind of mental gymnastics that you have to do in order to be an evolutionist these days. So here are 3 different kinds of freshwater fish. And almost all freshwater fish can be grouped as being part of one of these 3 families of fishes. As a side note, if you've never understood when to say fish and when to say fishes, there's actually a scientific rule about this. When you're talking about one species, fish is its own plural. So if I have an aquarium filled with a bunch of goldfish, it's an aquarium full of fish. When I'm talking about 2 or more species, fishes is the proper plural. So if I have an aquarium with goldfish and an algae eater, that's an aquarium of fishes. Okay? So here are 3 fishes, and all freshwater fishes can be traced back supposedly to one of these 3. All right? Well, what evolutionists wanted to do was they wanted to see how these three groups of freshwater fishes evolved from the saltwater fishes. So they did their phylogenetic analysis, they looked at DNA and so forth, and they came across something really interesting. According to their own analysis, almost all currently living saltwater fish evolved from one of these three groups of freshwater fish. Their own evolutionary analysis showed them the saltwater fish come from the freshwater fish. But of course this is a problem, because the geological column says, oh no, saltwater fish came first. The saltwater fishes evolved, then the freshwater fishes evolved from them. That's what the geological column says. But using their own evolutionary analysis, the evolutionists said, no. Freshwater fish evolved into saltwater fish. So if you're an evolutionist, how do you deal with it? Well, here's how the authors of the paper dealt with it. Our results suggest that ancient extinctions in the marine environment may have wiped out the earliest ray-finned fishes living in the ocean, and that the oceans were then recolonized from freshwater habitats, and that most marine fish species living today are descended from that recolonization. So if you want to continue to believe in evolution, here's what you have to believe. Freshwater fishes, or saltwater fishes evolved first. Then they evolved into freshwater fishes. Then most of the saltwater fishes went extinct. And those freshwater fishes evolved back into saltwater fishes. Isn't that wonderful?
Story for everything.
There's a story for everything. This is what, he was talking about how science needs to be falsifiable. This is why evolution is not falsifiable. You can make up a story to get around any data you don't like, including these. And this happens again and again in evolution. You've probably heard this before. This is from a PBS, this is from an episode of Nova. Today, many a school child can cite the figure most often called in forth in support of a common ancestor between ape and humans. Namely, we share almost 99% of our DNA with our closest living relative, the chimpanzee. 99%. Now, it turns out if you actually start reading a bunch of evolutionary sources, you'll find that that number varies. I've seen textbooks that put it as low as 95%, other textbooks that put it at 99%. But in general, the textbooks say we're somewhere between 95 and 99% genetically similar to our closest living relative, the chimpanzee. Now, before I get into this, I want to make a side note. We say our closest living relative is the chimpanzee because of the supposed genetic similarities. However, if we just look at our structural similarities, our closest living relative is the orangutan. If we just look at structural similarities, both skeletal and otherwise, we have significantly more features in common with the orangutan than we do with the chimpanzee. But genetically, we're most similar to the chimpanzee. So generally, evolutionists say that that's our closest living relative. I point this out because this is one of the biggest problems facing evolutionists today. And that is, do you believe structure or do you believe genetics? Because when I construct an evolutionary lineage from fossils, I get a completely different story than when I construct it from DNA. DNA tells a completely different story than the fossils. And if I look at things like RNA, I get yet another story. So in the end, our evolutionary lineages are hard to track down from an evolutionary point of view because it depends on whether you want to believe DNA, RNA, or body structures. But anyway, let's go back to DNA because that's what we're talking about here. So we're supposed to be 95 to 99% similar genetically to the chimpanzee. Here's the problem with all of these numbers. These numbers have been around since the 90s. The problem is we didn't sequence the chimpanzee genome until 2005. So prior to 2005, no one knew how similar we were to the chimpanzee because no one had completely sequenced the chimpanzee genome. Since we didn't know what the chimpanzee genome looked like, there's no way to really compare it to the human genome. Since 2005, people have been looking at the comparison between the human and chimp genome, and guess what? They're finding that there's nothing like 95, 99, 90, even 80% similarity. For example, in 2008, geneticist Richard Buggs over in the United Kingdom took the sequenced chimpanzee genome and tried to line it up as best he could with the human genome, and then count the differences. Here's what he says. The total similarity of the genomes could be below 70%. When he actually tried to directly compare the entire human genome to the entire chimpanzee genome, he couldn't find anything better than 70% similarity. Here's a more recent study that looks at it chromosome by chromosome. It takes the human chromosome and compares it to the chromosome that the chimp was supposed to, that's supposed to be similar to what's in the chimp. So chromosome by chromosome, it compares human DNA to chimp DNA. It sees the most similarity in a chromosome is about 75%, almost 80%. Some chromosomes, especially the Y chromosome, below 50%. On average, 70%. So when we look at these things genome-wide, we're seeing that humans and chimps are only about 70% similar genetically. Now this is a huge problem from an evolutionary standpoint because the lengths of these genomes are very large. We measure genomes in something called base pairs, and you don't need to know what a base pair is really, but humans and chimps have about 3 billion base pairs in their genome, all right? So a 70% similarity means a 30% difference, which means about 900 million base pairs are different. Mutations operate on a base pair by base pair framework. So in the end, we have to have 900 million mutations to get from the common ancestor to humans and chimps. According to the largest estimates, we only have about 6 million years to get there, according to evolutionists. So you need to have 900 million mutations fixed in 6 million years. That's 150 mutations a year fixed in the population. So the idea that evolutionary changes can actually produce this level of genetic disparity simply doesn't make sense. There's not the time, even under evolutionary circumstances. Probably the second most exciting thing that's been happening recently is the soft tissue found in bones that are supposed to be millions of years old. The story of this goes back 9 years to 2005, when Mary Schweitzer and her colleagues were trying to get a Tyrannosaurus rex femur out of a really tight spot. So the femur goes from the hip to the knee. That's a big bone. And it was in a place where they couldn't get it out whole. So they did something you never, ever, ever do. They broke it. Dinosaur fossils are so rare, they're worth their weight in gold. Breaking a dinosaur fossil is like breaking a Ming vase. You just don't do it. But they had to get it out. When they broke it open, they found this. Soft tissue. This is one clump of tissue, this is another clump of tissue. They're actually held together by an elastic piece. Wow. And they say that if you take tweezers and pull these apart and let go, come right back together. Wow. That's how elastic this is. All right, and this isn't the only sample, but that's the sample that one of the samples they highlighted because it was so soft and so elastic. Now, because it's in a Tyrannosaurus rex, the minimum age of this fossil is supposed to be 65 million years. Now, this is kind of crazy because, most soft tissue decays in less than a year, and even if I do theoretical calculations to find out how long the proteins that make up this tissue should last, most theoretical calculations say it's on the order of 30,000 years or so, even under ideal conditions. Yet here we've got this soft tissue in a bone that's supposed to be millions of years old. Well, obviously, at first evolutionists didn't like this, and some evolutionists still didn't like this, so they try and find out some other reason. For a long time, evolutionists were saying, what that is, that's not really soft tissue. It's a film left behind by bacteria who recently invaded the femur. Bacteria do leave films behind, we call them biofilms, and here's a picture, a microscopic picture of a biofilm, and you can see it kind of looks like that tissue before. Has a lot of the same properties of the tissue. So they said, that's what this is. This isn't tissue from the dinosaur, it's tissue from bacteria, or it's film from bacteria that, invaded the dinosaur bone fairly recently. However, Schweitzer and her colleagues actually did an analysis, a chemical analysis of the tissue, and they found a protein that is made by all vertebrates, all animals with backbones, but not made by bacteria at all. So it kind of makes sense that that was soft tissue. Since then, I think there have been so many examples found that it's hard to believe there's any other explanation. So for example, in 2009, Schweitzer and her colleagues found another set of soft tissue in a hadrosaur fossil that's supposed to be 80 million years old. Another group, headed by Maria McNamara, in 2009 found soft tissue in a salamander fossil that's supposed to be 18 million years old. In 2010, Johan Lindgren and his colleagues found soft tissue in the eye socket of a mosasaur fossil that's supposed to be 80 million years old. And when they looked at it under a light microscope, they found exactly the structures you would expect for eye tissue from a reptile. This is probably the most exciting one. It just came out last year. The fellow who published this paper was fired by his secular university after publishing this paper. And supposedly he's in litigation with the university right now over this. But basically, the author of this study and his colleague, they were looking at a Triceratops skull, and one of the horns, and it's not this skull, it's another one, but one of the horns on the skull had broken off. And since it had broken off, it wasn't as valuable, so they said, well, let's see what's inside. So what they did was they soaked it in a weak acid bath for several months. And what the weak acid does is it slowly eats away at anything that's mineralized, and it should leave behind anything that's soft. And so after a few months, they found all sorts of soft brown tissue coming out after the mineralization was eaten away. Now, of course, this soft brown tissue could be anything. Could be bacterial biofilm, for example. So once they got it out of the solution, they decided to look at it under a light microscope. Here's what they found. They found Haversian systems. Haversian systems are exactly what you expect from vertebrate bone. Vertebrate bone is composed of cells called osteocytes. And these cells have tiny little extensions that allow them to talk to the other cells. And those little extensions are called filopodial extensions. And in hard bone, they form these concentric circles, and these concentric circles are called Haversian systems. So this is clearly what you would expect from vertebrate bone. It's hard to understand how any other vertebrate's bone tissue could contaminate this fossil. So this is certainly from the vertebrate, from the Triceratops, that they were analyzing. Here's the amazing part. When they looked at it under a scanning electron microscope, they saw those same osteocytes again, but they looked specifically at the filopodial extensions. That's what's marked off in arrows here. These filopodial extensions, to give you an idea of how tiny and delicate they are, the length of this black bar is 20 millionths of a meter. All right? So these filopodial extensions are not even a millionth of a meter wide. Right? Here's what they say in their peer-reviewed paper. Filopodial extensions were delicate and showed no evidence of any permineralization or crystallization artifact, and therefore were interpreted to be soft. So not only is the tissue still soft, the dead cells inside the tissue are soft, and their tiny sub-millionth of a meter extensions are still soft. Do you really think that would stay soft for 65 million years? I don't see any possible way. It's surprising enough that it would stay soft for thousands of years. But this clearly shows that these bones can't be millions of years old. And to back that up, one thing that, he was talking about the white swan test. One thing science tends to do is blind itself. If it's not expecting a black swan, it won't go looking for it, right? Well, since dinosaur bones are all supposed to be millions of years old, no one ever expected to find carbon-14 in them. Carbon-14 is a radioactive isotope that even if I had a truckload full of it, after 100,000 years it would decay completely away to the point where there would be no way I could detect it. So since these bones are supposed to be millions of years old, evolutionists just always assume there's no carbon-14 in them. So why would he even go looking? Well, some creationists decided to go looking. And pretty much every sample that's been looked at has carbon-14 in it. So they've so far looked at 8 dinosaurs from all over the world, from Texas all the way to China and parts of Europe. And all of them have found enough carbon-14 in them that if you were carbon-14 dating them, they would date to about 39,000 years old, right? So now obviously we don't think that carbon-14 date is true, but the point is there should be no carbon-14 in them at all if they're even older than 100,000 years old. There's enough in them to indicate that under evolutionary assumptions, they're only 39,000 years old. Now, if you ask an evolutionist, where's that carbon-14 come from? They'll say, oh, it's contamination. Some modern carbon has gotten into the bone somehow, right? Now, of course, there were all sorts of tests and things they did to show that this is not contamination, but most evolutionists will have to say, well, this is just contamination. It's carbon-14 that got in later.
Isn't one of the biggest problems with their carbon-14 dating their assumption that there's no water element originally in the special, no nitrogen-14?
No, they don't use the amount of nitrogen-14. What they're looking for is the ratio between carbon-14 and carbon-12, and also between carbon-12 and carbon-13. So carbon-12 is stable, it doesn't decay, carbon-14 does. So if I look at, you know, something like me right now, the ratio of carbon-14 to carbon-12 in my body, and it would actually be this way, carbon-14 to carbon-12, the ratio of carbon-12 and carbon-14 in my body is indicative of what's in my environment right now. But if I were to die, I would stop exchanging carbon with my environment, so I couldn't replenish carbon-14. So the carbon-12 would stay the same, but the carbon-14 would start going down. Right? So it's only the ratio of those two isotopes that they use to determine. They don't use, because the nitrogen floats away anyway, so it's going to be hard to track that down.
Some people.
There are other like the potassium-argon dating system uses potassium and its Dalton product argon. And that's a problem because the argon's a gas. It can float away, it can do all sorts of crazy. It can also get in, back in. Gases can go in and out, right? Because it's pretty small compared to the holes in a rock. So in the end, I think that's a problem for potassium-argon dating, but not carbon-14.
Some people may have the misconception that carbon-14 gives you millions of years.
Yeah, and a lot of people think that. When you say millions of years, you tell people the Earth's only thousands of years old. One of the first things they often say is, what about carbon-14 dating? But carbon-14 dating can't give you millions of years.
Carbon-14 being the case young.
How many dinosaurs have they found with soft tissue ballpark you're aware of?
In the literature that's published that I've seen, I can count in my head 7.
Okay.
7 examples.
That's significant given the overall number of dinosaurs that they.
Yeah, and there are plenty where, you know, they went looking for soft tissue and didn't find any, too. But one would expect that. I mean, you know, the thing that's remarkable about the salamander fossil of all of those, the salamander fossil was muscle tissue, and that muscle tissue was laying on top of the bone. All this other soft tissue stuff was stuff you expect to have been inside the bone, and so be sort of protected. That muscle tissue was laying right on top of the bone and was still soft. That's pretty hard to understand.
I think Mark Armitage of the Creation Research Society is doing soft tissue analysis on.
Mark Armitage is the fellow who did that Triceratops. He got fired from his position right after he published.
We'll do one more question, then we're going to do more at the end, but I've got about half an hour. We're going to walk you through these various people that are going to be coming, and one example from each of their areas of science. So we'll wrap it up with one more question, then we'll come back to it later.
See, now you've scared me. Oh, there you go.
Have you learned about or researched the BICEP telescope?
Oh yeah, the BICEP results that have come out really recently. If you don't know what he's talking about, in order to force the Big Bang theory to be consistent with the data at hand, one of the things that the Big Bang has to incorporate is a process called inflation. This is a process that we have no idea of how it would start. Once it starts, we have no idea of how it would stop. But it must have happened in order for the Big Bang to have worked. And basically, during the period of inflation, the universe expanded at hundreds of times the speed of light. And this gets over a couple of problems that I don't have time to get into. Well, of course, since it's such an odd thing, we don't know how it starts, we don't know how it stops, and all of that, people who believe in the Big Bang really want to confirm it. Well, one way they try to confirm it is, if they look at this background radiation that's coming from all of space, the process of inflation is supposed to have warped the light so that it tends to vibrate one way and not other. So we call that polarized light. So we expect the cosmic microwave, the background radiation from the universe, to be slightly polarized if inflation occurred. And just recently, a report that was out from the BICEP team that says they've been looking in this part of the universe that supposedly has no magnetic field problems, very little dust and so forth, and they think they've seen this polarization. Here's the problem. Magnetic fields, gravitational fields, and electromagnetically charged dust will all produce this polarization as well. So what they've had to do is they've had to point their telescope in a direction where we think there's none of that stuff going on. But even there, we know there's going to be some dust along the way, we just don't know how much. So in their scientific paper, they actually say, well, here's how much dust we think is there. We'll subtract the effect of that dust out, and look, there's still some left over. Well, we don't have any measurements to tell us how much dust is out there. So this estimate they haven't done is really truly just pulling a number out of the air. And if their estimate is off by even a factor of 3, which sounds like a lot when you're measuring something with a ruler, but when you're just taking a guess, even if their estimate is off by as little as a factor of 3, then the effect they see goes away and it's all accounted for by the dust. So in the end, I think it's this real desire to prove this physically unrealistic thing called inflation. And they're putting way too much emphasis on the results. Twice before, people have thought they've seen this. And twice before, after about a year, it was shown they didn't see it at all. I expect that's what will happen here. But you won't get the media attention to the fact that it's been shown to be wrong. You won't get the media attention that you see for this supposed discovery.
Thank you, Jay. Sure. Now, you start to get a feel for the quality of things that folks are going to see here in the fall. You know, they start to hear this sort of stuff. All of a sudden, the fact that the box is a box maker becomes real in these kids' minds. And I'm going to get at the end of this next half hour or so about why can't people see the obvious? Because I've struggled with that question for 20 years. By the way, I came out of college just like Jay. I would have called myself an agnostic versus an atheist, but totally convinced that everything can be explained without God. Now I'm going to walk you through, as I said, the speakers that are coming. I'm going to start with the first one, Dr. Donald DeYoung. Some of you have heard of him, some of you haven't. But Dr. DeYoung, he's the chair of the Science and Math Department at Grace College. His PhD is in physics. He's the president of the Creation Research Society. Now there are 1,700 members. Don't ever let anybody get away, especially students, with it being implied or said that if we believe the box has a box maker, if we believe in a creator, if we believe in a recent creation, science will come grinding to a halt. All of the founders of modern science, in essence, maybe there are a few exceptions, they believed in a literal recent creation by a creator. Isaac Newton and Michael Faraday and James Maxwell and Lord Kelvin, all these guys, Pasteur, they believed in a recent creator and they set the very foundation of modern science in place. So you've got to separate the operation of the world, which we've gotten very good at figuring out, versus the origin of the laws of science and the origin of things. And that's just glossed over as if the operation explains the origin. And he's authored over 20 books on the evidence for creation. Now his expert is really cosmology and physics and so on. One of the subjects he's going to be talking about is the design in biological creatures that allow us to develop new technologies and new products. What's that, Jay? Mimicry? Mimicry. Yeah, mimicry is how you pronounce the word, thank you. The other will be on the origin of space. Now I'm going to take 5 minutes and just give you a feel for the kind of things kids are going to be shown that they never get to hear about. It never shows up in their classrooms. The opposite shows up in their classroom, and that is, I'm going to talk about the origin of stars for just a moment. Now, you will see in all the Cosmos specials and the textbooks charts that look like this. And when I see them, they kind of remind me of the evolution charts where they line up animals. They line up the critters to try to explain how one animal turned into another that turned into another by lining up similarities. They'll line up big gas clouds and assume they turn into big stars, and little gas clouds turn into smaller stars in different colors and different sizes, and quasars and pulsars and all this cool stuff out there. And it all sounds like it's been explained, all right? And you'll hear it implied that it's all been explained. Well, here is reality that the kids don't get to hear about in their classes. First, just a comment. You don't see order coming about from explosive expansions. You hear the Big Bang, they say, oh, it's not an explosion, it's just an explosive expansion, rapid expansion. In essence, it's an enormous explosion or everything that came from nothing, but they don't want to talk about that typically. But the fact is, we do not see stars forming. And here's why. I'm going to give you 3 reasons. First is that there's a law of science called the natural gas law. Now, a law is a law of science only if there's never been an observable, reproducible experiment or example or observation that shows that it's in doubt, that it's downgraded to a theory, all right? So a law means we've never seen an exception, all right? This is still a law of science. And in essence, you hear it's PV equals nRT, you've all heard that. But one of the things that says is that gas will always, left to itself, move from an area of high pressure out to an area of low pressure. Because the gas molecules are bumping into each other. They're shoving each other apart. Now, outer space is a vacuum where the gas molecules are really far apart. But let's just look at this room. I've got a can here where the gas molecules are packed tightly together, high pressure inside of this can. In this room, the gas molecules are farther apart, low pressure, all right? The only thing separating high pressure from low pressure is this little valve. Now, for a star to form, you'd have to have gas molecules far apart, packed tighter and tighter and tighter and tighter and tighter and tighter and tighter together. Trillions of times tighter than we find them in outer space, until they're so packed together, they burst into nuclear flame and nuclear fusion starts to take place. Well, that's like all the gas in this room when I open the valve, deciding I'm going to pack myself tighter and tighter into this can. Now, I like to do experiments. That's what science is all about. So I'm going to open the valve, and because we're all told in all the textbooks and all the Nova Specials that stars are forming out there all the time they're forming, that means maybe the gas in this room is going to get sucked into this can. So you better hold your breath. 3, 2, 1. Which way did the gas go? From high pressure out to low pressure. Every single time. Never an exception if left to itself. See, that's what science shows us. And then, turns out, when things are moving, if there's no restraining force, a race car going around a track, the track is sloped and there's friction with the tires, but if those things weren't there, as it started to go around the racetrack, it would go shooting outwards. In outer space, gas is swirling. There's no restraining force because the gravity is way too small, and yet stars are spinning. The gas should have spewed outward, not condensed inward. And lastly, the particles needed to make a star are charged. So it'd be like putting a bunch of positive magnets together. See, the gas law, the momentum laws, the charged electrostatic energy laws are 1 billion times stronger than the gravitational pull of the molecules. All of these things say gas is never going to collapse to form a star. And what are we told? Stars are forming all the time. Now, why do they say that? Because they're stuck with believing the box made itself. They can't separate the operation of things from the origin of things. You see, stars are burning up their hydrogen, and there's not enough there for them to be as old as they need to believe the universe is. Therefore, they have to believe stars are continually remaking themselves. Now, if you look at the universe, we think there's about 100 billion galaxies out there. If they're each kind of like the Milky Way, it has, we believe, about 100 billion stars. That's 10 with 22 zeros. You divide that number of stars by how old we're told the universe is, it means every second there should be 20,000 new stars forming. Next second, 20,000 stars. Next second, 20,000 stars. We've looked at every little bit of outer space with the Hubble Space Telescope, in essence. And then we'll come back 10 years later and we'll look at that same spot, blow it up so we can actually count stars in faraway galaxies. And we've never once seen a new star form. The laws of science say they're not forming. Observation says we've never seen one form. What conclusion should we come to as a scientist? Something different must have happened in the past. See, I think God put the laws of science in front of us so we would know the origin of things is different than the operation of things. Is this rocket science? I think it's obvious. But who's going to give it to the kids? That's our job. This is important. It gives them reasons to have credibility for understanding the box as a box maker, and then start to explore who is this box maker. Now, just a few quotes. The origin of stars is one of the most fundamental unsolved problems of contemporary astrophysics. Those don't make it down into their way of most textbooks. They occasionally come up in these scientific papers, and then they get glossed over. Their answer is typically, well, maybe we'll figure it out in the future. That's not science, that's faith. If stars did not exist, here's another researcher, it'd be easy to prove you never expect one to exist. Most of us are persuaded stars form from diffuse matter, gas clouds, that has to condense and contract. Nevertheless, nearly all observations, and by the way, it's probably all observations, of these gas clouds show it's flowing outward, not condensing inward. We start from a shaky foundation. We can't explain a single star, yet we have to explain 10 billion of them. If none of us knew stars existed, all of the research, frontline research, would convince us a star could never form. See, that's all I've shown you. But this stuff doesn't get out in front of students for the most part. And last, Stephen Hawking, the supposed greatest physicist of today, in his book, A Brief History of Time, he said, the Big Bang basically explains everything. But just to show that just because someone is incredibly smart doesn't mean they're right. On the same page, he says, well, there are a few problems, such as the origin of stars and the origin of galaxies. We don't know how either one formed. Well, if you can't explain stars, you haven't explained planets, and you haven't explained larger chemicals, and you haven't explained life. In essence, you haven't explained anything if you can't even explain where a star comes from. That's the kind of stuff we want to get in front of the kids. Very credible for people who have studied it and know the facts. See, this isn't a little side issue. This is a foundational black swan that looks at the very way the laws of science operate so that we will know what the truth is. Either the box is a box maker or the box made itself. The laws of science say the box couldn't have made itself, starting with what we know about stars. And you do the same with time, energy, and matter. Energy and matter can never be created or never be destroyed, then where did it all come from? The box has a box maker. Second person coming, Dr. John Sanford. Dr. Sanford worked at Cornell University, major United States university, for 34 years. His PhD is in genetics. He developed what is known as this concept of genetic entropy. After about 30 years of working at Cornell teaching genetic biology, he's realized it can't possibly be true. I'm going to show you why in a second. He realized all genetic codes are deteriorating downwards. None of them are increasing in useful, functional complexity. He's published over 100 scientific publications. He's multiple patents. He invented the technique for taking pieces of DNA out of one type of plant and implanting them into the nucleus DNA of another plant in order to do crossbreeding of genetic information. He worked extensively for the Institute for Creation Research. Brilliant scientist, highly esteemed in the entire intellectual community. I mean, we can't pay these people a fraction of what they're worth to come to this conference. Now, they'll be compensated because they refuse to allow people to come without any compensation, but they're coming because they love the truth and they want the students to hear it. We still got to fund this thing, all right? This first section is about genetic deterioration in the citric-eating bacteria. Huge splash. I believe it came out of MSU, where they bred a bacteria for 30,000 generations, and finally it started, you saw the news article said, bacteria evolves to eat plastic. What he's going to talk about that has nothing to do with evolution. It actually is a breaking down of the mechanism that bacteria originally contained. And then he's going to talk about, and I got to read this, mutations and useful genetic information. Now, one example, well, two examples. We all hear mutations are what drive it all, okay? Mutations result in a loss of functional information. The best example I've been able to come up with is a flash drive that people really can connect with. Used to have all my lectures on a flash drive, paid $59 for a 512 megabyte flash drive. How about that? 512 megabytes. Because it was one of the first ones out. Now you guys are laughing at that because you know they've got like 64 gigabyte flash drives for less than that nowadays. But I'm cheap and I ain't about to go out and buy a new flash drive. when I can just add a mutation to my flash drive and make it work better. So I added my first mutation. Now that is a pretty good representation of what a mutation is. It's like a cosmic ray coming down from the sun and blowing apart some of the coded information of DNA and rearranging it. Okay, it's like driving a nail through that thing. Now maybe that nail could connect a couple circuits that didn't used to be there. Maybe it might change the function of that thing. But it's not going to increase the total amount of functional complexity. But so being an engineer, I tested it, plugged it in my computer, and then added more mutations, and I tested it, and added more mutations, and tested it. See, it's almost laughable. But how about if I wanted to develop a school system where every child in a country could only be allowed to hear that the origin of flash drive came about by people driving nails through random pieces of electronic circuitry? You'd lock me up and put me in an insane asylum. But that is our educational system. It's the only thing they're allowed to consider. Natural random changes. Now here's what Dr. Sanford brought to this field. All right? When I was young, and it's what about natural selection? But if those changes have some sort of an advantage, all right? It doesn't have to be an increase of complexity. Something changes to give it an advantage. Then that creature's going to make more copies. You do that millions of times over billions of years, surely some creature could turn into a different kind of creature. And that's the kind of things kids are going to hear. If that's all they hear, it's going to sound logical to them. We have to have answers. This is Dr. Sanford's contribution, which is just phenomenal. And it's his example out of his book called Genetic Entropy. Charles Darwin is called the greatest scientist of the 1800s because he came up with the idea of natural selection. Well, let's look at natural selection like we look at a wagon manufacturing plant. You know, when I was young, I had a little red wagon like this. I loved my red wagon. I would ride around it when I was 4 and 5 and 6 years old. And when I got older, my friends got bicycles and tricycles, you know, moved to motorcycles and cars. But when I was 16, I'm still riding around in my wagon, okay? Because I'm a dreamer. I don't just want a bicycle. I don't just want a tricycle. What I want is the Starship Enterprise. Now, the difference between that wagon and the Starship Enterprise is like the difference between a bacteria and a human being. Think of all the parts you have to add. You got to add information that will make bones and muscles and hearts and livers and brains, and all this stuff has to work in perfect concert. How's that going to happen? Well, this is what we're told. The wagon comes off the assembly plant. The quality control department looks at the wagon, and it says it's the right size, it's the right shape. Off it goes to market to see how well it will sell. Natural selection looks at an animal, it has offspring, makes a copy of itself, looks at these new the next generation, they're the right size, they're the right shape, off they go into the environment to see how well they survive. If they have an advantage, they're going to survive better. Then they'll make more copies of like themselves. Well, except what we now know is that underneath the wagon, and let me just move some stuff around here, underneath the wagon is a book. It's the plans for building the next factory to make the next wagon. Every time life makes a copy, it takes the DNA code, copies the DNA code, and rebuilds the entire factory to make the next animal. Okay? That's what we do when we have a next generation of human beings. Starting with chemicals, the information on DNA pulls them together to form the cell to make the very next human being. And every time that happens, the information inside of that book gets randomly changed. Just a little bit. So if this wagon is really simple, let's say this book only has 1 million letters. The DNA code of a bacteria may only have 1 million letters. So I'm just going to change one letter here, and then I might turn over, say, 10 pages, all right, or maybe 100 pages, and I'll change one letter on that page, and I'll turn over another 10 pages, and I'll mark out one word on that page. And let's say I just do that with 10 words, 10 letters. Now I take that book, build the next factory, and make the next wagon. Do you think that second factory is going to be able to make that wagon and it's going to function properly? If I change 10 letters out of 1 million letters, it's like one letter out of every 100 pages. If you read that book, do you think you'd be able to understand that book? What do you think? Yeah, you would. You might not even notice it. But the third wagon has 20 changes, random rearrangements. 10 wagons later, you have 100 random rearrangements. 100 wagons later, you have 1,000 random rearrangements of the letters. 1,000 wagons later, you have 100,000 random rearrangements of the letters. What do you think is going to start happening? Wheels are going to fall off. The quality control department will see that wagon, they'll kick it aside. But the wagon right before it also had those 1,000 random rearrangements, and it might have survived, but it took with it that ever-increasing little more mistakes, little more mistakes. See, the quality control department can't see the book, and it can't change the changes in the book. The natural selection can't see the DNA code. It can't stop this slow, gradual, itsy-bitsy, teeny-weeny, downward deterioration of the coded information. There are now 4,000 known genetic mistakes, random mutations to the human genome. This is what we now know. See, there's a quote that says if there's even one deleterious negative mutation per generation, it will doom any species to extinction. Before we knew what was happening, every human generation, there's a whole bunch of different kind of random rearrangements, random rewriting of the book, which will, when you add them up, you get somewhere between, your children are going to have somewhere between 1,000 and 10,000 random mistakes to their DNA that you didn't have. And you have somewhere between 1,000 and 10,000 more random mistakes than your parents had. That's why early on human beings lived for hundreds and hundreds of years, and now we don't, because it built up and built up and built up until it causes major problems. That's why there's 4,000 known genetic allergies, problems, autism, and all sorts of diseases tied to mistakes on our genetic code. Nothing could survive this for millions of years. The man realized that, he gave his life to the Lord, he became a Christian, and he's doing everything he can to help students understand what's really the reality of science and history. See, it's not, now what they'll say, well, you know, maybe the word blue or red got changed to blue, and the blue wagon would sell really well. A positive mutation, not an upward increase in complexity, okay, just a change. Well, that blue wagon would take with it all of those thousands upon thousands of little tiny errors that would then continue to build up and destroy ultimately all the information. See, that's what students need to hear. They need to hear why evolution could not possibly be true. And it's a reality. Enormous black swan. Who's going to let them see the black swans? We are, by bringing this conference to MSU. Third speaker, Dr. Jerry Bergman, college professor for 34 years. He was fired from Bowling Green University in the 1980s because as a biology teacher, he was pointing out problems with evolution, removed from his job. And he was told he was doing it because he was doubting evolution. Sued the university, said this is discrimination. The judge in the case just threw it out and said, the schools, they need to teach naturalistic stuff. Anything else is religion, removed from his job. But he's continued to teach. He has 9 advanced science degrees, 9 master's degrees in every area of science, from geological sciences, genetic sciences, biological sciences. His PhD is in human biology. He's published 700 papers out there throughout the technical scientific community and the Creation Society, written 20 books on the evidences for creation. And he's one of the top IQs. I think the top 1% of the population can join this MESNA organization. Brilliant man. He's coming here to teach our kids what the truth is, all right? His topics are going to be censorship in science and why Darwinian evolution is wrong. I gave you very general topics so you can go whatever direction you want with this. Just one quick example, because I need to move on. This experiment is shown in every biology book in the country, in the world, to try to explain how life could have made itself. It's the idea that the building blocks of life could form randomly by random energy input. And just a little bit of background. Our bodies are biological machines that contain a soul and a spirit, but we are biological machines as a human being. The reason we don't live forever is because every time we have a copy, every time our cells make a copy, there are copying mistakes, okay? Originally, it wouldn't have been that way. We really, literally would have lived forever had God not brought death into the universe. Well, the most common molecule in our body is a protein, and there's hundreds of thousands of different shapes, arrangements, sizes of these proteins that act as parts of, like motors, parts of they carry things, they act as cell membranes. Our muscles is a certain protein. Hemoglobin in our blood that carries oxygen is a certain protein, and so on. The proteins are like a long beaded necklace like this that are just linked together, that it bends and twists and folds in just a certain way to form certain things, functions in our cell. Well, the beads are called amino acids. This experiment done in 1952 showed that these amino acids can form by themselves if you just add energy to them. Ammonia, methane, hydrogen, and water. You add a spark of energy, you can get these beads, okay? The textbook then goes on to say, see, this shows that life could have formed itself. All right? And it is laughable because this is like stepping on a sheet of paper and saying, now I've climbed Mount Everest. That's how close we really are by showing these amino acids could form. But it gets worse than that. Now remember that date, 1952. This is still in every biology textbook in the world, all right? Keep this thought in mind. Don't you think that if they'd come up with something better in 62 years, that's what would be in the textbooks? This is the best they've got in 60 years. They left oxygen out. Typically doesn't even get mentioned, or lots of times it doesn't. Yeah, every time we go down the rock layers, we find oxygen, far down as we go. There's no evidence whatsoever oxygen hadn't always been around, and there's lots of evidence that it has always been around. If you put oxygen in, you don't get amino acids. You only get a few of the different colored beads, maybe 6 to 8 of them. And yet every form of life has 20 different specifically designed, specifically shaped amino acids. Every time this experiment's done, you don't get them. You only get a few of them. Honestly, it's never been shown where the other one's going to come from. These very specific, very complex biological molecules only are made by life. If you don't have life, you don't get the right molecules. You don't find them out there in nature. The energy input would be destroying everything else. You get this 50-50 mixture of like right and left-hand gloves every time the experiment's done. And yet because God wanted us to absolutely for sure know what the truth was, that the box had to have a box maker, every form of life only uses the left-hand glove. And yet the drawer is filled with a mixture of both. And they spontaneously will link together. Nobody's ever shown how that could happen. You would get the Nobel Prize in Science if you could show how it happened. But you can't, because it doesn't happen. God wanted us to know the truth. And then if you look at one of these molecules, by the way, this is kind of a bottle of a protein molecule. Kind of a long one, but nevertheless, there's thousands of them that are this long. If I was to just change one blue bead with one red bead, one swap of one color, it would be a disease. If this were hemoglobin, it would be a disease called sickle cell anemia. And it wouldn't bend and fold quite right, and it wouldn't carry blood very well, and most people would end up dying from it. Because of one colored bead in the wrong place, one wrong amino acid. I think maybe God wants us to know the truth. That's the kind of stuff people need to be exposed to. I'm going to jump past this. See, even if you have all the components, and we've done this experiment where we put every chemical you need for life in exactly the right spot, and it's in the right concentration, and it's in the right location, and we put it in a container where heat or light can pass in and out. And then we open the container to see if a new form of life can come out. And we've run this experiment about 10 billion times last year. It's called canned food. It's all the chemicals you need. Right amount, right concentration, right everything, they're there. And yet we never open a can of Dole pineapple when a little blue alien creature crawls out. See what the truth is? Enormous black swan. It can't happen by itself. Okay, now last speaker, Dr. John Whitmore. He's a professor of geology at Cedarville University. He's got a master's in geology, a PhD in biology, but with a paleontology emphasis. Once again, rock layers. He's a rock expert. He's written multiple papers and publications. He's been to Alaska to do dinosaur expeditions, written a book called The Great Alaska Dinosaur Adventure. And he's worked with the U.S. Geological Survey, active member of the Creation Geological Society, the Creation Research Society, because you've got to put this creation of life in a real time frame of Earth history. If you don't do that, it's still not credible to the people around us. So he's the one who's going to talk about the rocks and the fossils and so on. Now, one more example that I'm going to wrap up with why As obvious as the kind of stuff I've shown you, why will people who see this kind of stuff be blinded to it, even when it's right in front of their face? But this is what goes on in the rock layers. And let's just give an example of all these skulls we find. The kids are convinced that apes have turned into people because they lined up the skulls. Well, let me take a set of skulls, all right, and show you the kind of thing that honestly, literally, really does go on. Suppose I'm a paleontologist and I find this critter in the rock layer, okay? He's a pretty vicious guy, big teeth, okay? So I'm going to immediately start speculating. I wasn't there to see him live. I didn't see the rock layers get buried, but I found this guy. Well, I'm going to assume he's vicious. I don't want to meet him in a back alley. He's probably evolved for something else that wants to look sort of like him, and I put him on a shelf, give him a scientific name. Well, then suppose I find another one that's very similar in shape. Now you'll notice they're different sizes. And I don't know if you can see it, but this one has a single ridge down the top of his head. This one has two ridges, one down each side of his head. And I find a bunch of them, so I know he's full grown. And I'm thinking, where did he come from? Might be a higher rock layer, might be the same rock layer. I've got to explain it somehow. I mean, if God didn't make these creatures, then they must have somehow one turned into another. It's the only possibility. It's one or the other. And I'm telling you, there is literally hatred for someone who comes on a college campus and tries to explain that there's a box maker that made the creatures. There's such of an atmosphere of antagonism. We gotta do it anyway. We gotta do it, because it changes lives. But I'm gonna make up a story. I'm gonna say, well, maybe this guy had different offspring because they're smaller, they've got to hide, they gotta be faster. So maybe this one lives in the forest and this one lives where there's not so much forest. And because he's smaller, he's faster and he has to run around, but it's just a slight variation that happened as one creature turned into the other. So then I find another skull, let's say I find this skull, and you'll notice the jaw is sticking way out now, and the forehead is getting higher, all right? Same general shape, but significant differences. How can I explain it? Well, maybe this guy, because his brain is bigger, he's now learned he can dig underneath the roots and he can grab animals and grubs and stuff, and he can use these big teeth to dig with. So he's like, can both kill animals and eat them, but he can also use his body to find other sorts of creatures that he couldn't otherwise find. But then I find the Einstein of this kind of creature. Look at the domed forehead on that little guy. He must gotta be smart. I mean, look at his brain. Not only that, but this guy wouldn't even make a good snack for the original one. So he's gotta be smart to escape. And he probably runs around in the forest and he uses his very big brain in comparison to the size of his body, probably 2 or 3 times brain to body mass of the original creature. And he evolved these abilities to survive in spite of his much smaller, limited size, and then found an environmental niche. And you see what I've done? I've explained how those creatures could have developed by slow, gradual changes over time. And it's going to sound really logical to the kids if that's all they hear. If evolution's a fact, evolution's a fact, evolution's a fact, millions of years, billions of years, billions of years, billions of years, from the time they're 3-year-olds watching dinosaur films till they're in college, everything gets filtered through that. You go out and find a series of bones, you're going to line them up. It's just logical. And it's going to sound logical, except it's all wrong. Everything I just told you is totally wrong. See, the first creature was a gray wolf. And because we can pull apart the genetic code, we now know every kind of modern dog came from the coded information that was already on the gray wolf. No evolution happened. They call it evolution. It's just a sorting of information that was there. The second creature is a fox, variation of the genetic information already on the wolf. The last creature was a chihuahua. It wasn't the Einstein of the canines. You take dogs and you breed together the smaller ones, and you breed together the smaller ones, you breed together the smaller ones, you eventually end up with this little worthless rat-like creature that wouldn't survive for 10 minutes out there in the wild. It's not an Einstein. It's just you've gotten rid of everything that didn't look like a wolf and was useful, and you end up with a chihuahua. And the fourth one was a bulldog. See, they all came from the wolf. No evolution happened. That's what goes on in the rock layers. That's what goes on with misinterpreting things. Because if you don't use God's word as your guidebook, And 10 times it says creatures reproduce after their own kind. Trees make trees, fish make fish, birds make birds, cattle make cattle. It says it 10 times in 10 different ways. You're going to misinterpret where they came from. See, it's a biology book. All right, now I'm going to jump on ahead. If there's a debate, Dr. Charles Jackson will be joining us to do the debate. He's done lots and lots of debates, speaks at Hilldays College, wins them essentially every other time. Jay would do them if he was here, but there'll be more opportunities. And I'm going to end up with this. Let's just look at one creature God has made. And I do this one just because I'm an engineer and it blows me away. You know, there's about 20,000 species of these things called leafhoppers, all right? Now, that doesn't mean God made 20,000 different kind of creatures. It means God, like the wolf, put a ton of variety built into the genetic code of the original leafhopper. He wanted to fill the environment, he wanted to show his creativity, and as there are advantages of different colors, different shapes, different sizes, then they do spread out across the globe. That does happen. They call that evolution. It's really just variation within a kind. Now, this guy has hind legs like nothing you can imagine designed for jumping. I mean, he puts the grasshopper to shame like crazy, okay? He actually accelerates at 100 g-force of acceleration when he takes off. Now, test pilots, when they're zipping around in a fighter jet and going into a sharp bank turn, when they hit about 10 Gs, they're in danger of blacking out. And they put them in pressure suits to force blood back up out of their extremities. Now here's a centrifuge where they test these things. I want to show you what 10 Gs looks like if someone's sitting in one of these spinning around centrifuges. Okay, there you go. That's what 10 Gs simulated looks like, okay? It's just a lot of force. Well, this leafhopper, when he jumps, the little nymph, he takes off at 100 Gs, all right? It would blast our body apart, all right? Now here's the problem. His legs move so fast that the nerve signal, the way it's wired, can't get to both legs simultaneously. Now think about this. It's getting ready to jump. It takes off, but one leg moves faster than another, so it careens sideways and slams its head into the side of the tree, into the creature. How do you solve that problem as a creator? All right? All right, now wait for this. I love it because I'm an engineer, okay? This is what God put, connecting the legs at the base of the leafhopper so that when one started to move, the other one had to be forced to move in concert. It's an electron microscope picture. Are you ready for it? Of what's at the base of this little insect. There it is. Is that unbelievable or what? How do you go into a machine shop and randomly pound hunks of metal and come up with a pair of intermeshing gears that perfectly fit and coordinated together? How do you make one little teeny change of molecules that have to be useful and functional or the creature would go extinct? And you need thousands upon thousands of changes to have created those gears.
Hello, this is Mike Smith with Creation Summit. And as Bruce Malone just shared, we are co-sponsoring the November 1st Creation Conference on the MSU campus. This is a one-day event. It's open to the public and it is totally free. For more information, just go to creationsummit.com and click on the MSU link that's found at the very top of the page. Hope you can make it. God bless.